Okay, welcome Year 12 to the very first edition of Mr. G Does Biology. Unusual for me to be recording this, so if I go wrong, apologies, I'll try and get it right. If you've got any questions, don't be afraid just to email me and I'll try and address anything that I maybe haven't explained the way I normally would. You need a pen, bit of paper, and hopefully you'll be able to follow everything as we go through. You will need to pause this video from time to time when I give you particular tasks to do, and hopefully you'll have no problem following on and almost treating it like a normal lesson. If I'm not looking at the screen, it's because I'm looking at the keyboard or the tablet I've got in front of me to annotate things, and we'll just see how this goes. Okay, first thing we're going to do is just do a little bit of knowledge retrieval. You just start the same way every lesson, so let's keep it the same. And so your first task, fairly straightforward. What I'd like you to do, looking at the DNA sequence that will appear on the screen, could you please write down what you think the messenger RNA sequence would be? Off you go. I recommend you pause the screen while you write this down, and then I'll reveal the solution shortly. Okay, hopefully you've managed to, excuse me, hopefully you've managed to come up with a solution to that. Here it is. Key thing to remember is the incorporation of uracil as we produce our messenger RNA. So we have our thymine to adenine, adenine to uracil, and so on. And if you've managed to get that particular messenger RNA sequence, well done, you've remembered something from last lesson. So let's quickly recap what we've looked at in the last couple of lessons. And so we started off looking at the structure of DNA. We then moved on and went through the process of transcription, which was how we produced our pre-messenger RNA. Remembering here we're looking at our eukaryotic cells rather than our prokaryotic cells. And we then moved on. Once we have our pre-messenger RNA, remember everything's currently in the nucleus. We're then going to go through and look at the next stage, which is splicing. Splicing enables our non-coding introns to be removed and our exons to be joined together. This is still within the nucleus, and we're then going to go on and look at how we can produce our protein in our translation process, which is now going to occur at the ribosome, and we now have our sequence of amino acids, and we can produce our functional protein. The thing we're going to look at today, though, is looking at when some of this process goes wrong and what the possible consequences are. So if you understand really well kind of what's happening in our production of our amino acid sequence, it shouldn't be too far for you to realize that if anything goes wrong in the production of the pre-messenger RNA or in the splicing stage, it's going to have consequences for what is translated in terms of our amino acid sequence. So let's have a look at what we mean by the term mutation. Mutation has a specific definition that you should try and recall because it's defining a particular event that's taking place. And what we need to look at, it's to do with the arrangement of bases in an individual gene or the structure of a chromosome. It isn't what many people think. It's not this, for example. It's not the science fiction side of it. We're focusing purely on what's happening to the particular bases in our DNA. So what might the consequences of that be? That's what we're going to have a look at. However, as mistakes do occur, there's obviously things that can happen to try and correct those errors. So within a cell, there are opportunities to repair mutations. So mutations can occur in any cell, whether that's the gametes, e.g. the sex cells, but it can also occur in somatic cells, that's any other cell within the body. And depending where the mutation is, it can have particular consequences in terms of evolution or passing on that mutation to a future generation. There are specific enzymes that can repair faulty DNA and make corrections to any errors that can occur. And if we don't have any of these mutations corrected, it can lead to significant problems with the production of new protein, and that's what we're going to focus on in particular. What happens when our unrepaired mutations go through the translation process and what those consequences might be. So let's start with looking at perhaps the easiest type of mutation, which is our substitution mutation. We're going to focus on just one single base change. You can get multiple base changes, but just to keep things simple, we'll just look at one and look at the consequences. So if this was our particular coding sequence, and this is what we have in terms of producing our amino acid sequence, what we would need to think about is the consequences of a particular base change within our coding sequence. So we have our sets of 3 nucleotides coding for particular amino acids. Let's look at what consequence might be if there is a particular mutation, an error that's occurring. In this case, we're looking at a substitution. So as the name suggests, substitute, we're changing one particular base for another. So let's look at the consequences of that if we do have one particular base change. So here we're changing that particular base, our adenine to thymine residue, and the consequence of that particular base change is quite fundamental. We've now got a different amino acid that has been coded for. So our amino acid sequence that we produce now is no longer the same as it was before. So have a think what the effect of that might actually be. I don't mean that particular change directly, but any type of substitution mutation. What kind of effects do you think might occur with any substitution? So pause the video, have a think, and then we'll go through the examples that can occur when we've got a substitution mutation. Okay, so let's have a look at what can happen. We get something as straightforward as what's called a nonsense mutation. A nonsense mutation is quite simply if there has been a base change, but if you think of your genetic code, what we've coded for is a stop codon. So if we've coded for a stop codon, clearly polypeptide synthesis is going to stop, and so we're not going to produce our functional protein. It's going to end when we reach that new stop codon that our mutation has led to. We've also got something called a silent mutation. A silent mutation occurs when we have the same amino acid that's being coded for, so we don't actually have an effect in terms of the polypeptide sequence, although our DNA sequence or our messenger RNA sequence has been altered. Remember, this is due to the degenerate nature of the genetic code. We've got multiple particular codons coding for a particular amino acid, and so it may be that that mutation has led to just one change in one of the bases, and we don't change the amino acid that is coded for. Okay, so the next example to look at is our missense mutation. With a missense mutation, it's a fundamental difference in that we're going to end up coding for a different amino acid. If we code for a different amino acid, that can have a dramatic impact on the final tertiary structure or quaternary structure of our protein. We'll look at a particular example. In this case, the example we're going to go for is glutamic acid being replaced by valine in the condition sickle cell anemia. Sickle cell anemia is a disease of the blood, a genetic disorder that leads to our hemoglobin changing its shape. Normally hemoglobin has our quaternary structure, but what can happen with our hemoglobin in this scenario is, but let's zoom in and have a quick look. You can see normal hemoglobin is this kind of structure. However, with this mutation, you can lead to clumped hemoglobin, where it's forming kind of rod-like structures. Ignore the spelling, it's just the image I got it from. Hopefully you know how to spell hemoglobin correctly. So hopefully I'll watch this little video clip, but the basic consequences of that mutation are quite simply a change in the shape of the red blood cells. So they normally should be this biconcave disc, but they can form these sickle cell shaped structures, and the sickle cell shaped blood can get stuck in capillaries and small arterioles and venules, and that can lead to a problem in terms of oxygen transfer to surrounding tissues, and significant issues in terms of respiration within those cells. So let's watch a brief video clip. If you want to go to this website yourself, just pause the clip and type the website in, but we'll try and play it and record it and see how well this works. It might not, we shall try. Okay, let's maximize the screen. Let's have a look at the clip, and hopefully you'll hear the sound as well.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under certain conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single code letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in the position where glutamic acid should be. The valine makes the hemoglobin molecules stick together, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
Okay, so as you can see, quite a significant impact leading to a well-known genetic disorder. So those are all the effect of a substitution mutation. We can have others, so we'll have a look at one or two other examples where we're not looking at substitution, but we're looking at things like deletion or insertion. So the name kind of suggests exactly what's going to happen. With deletion, we're going to remove a particular base from our sequence. With insertion, we're going to add a particular base. The consequence of this can also be obviously quite dramatic. So let's have a look at what happens if we move through. So in this case, that particular base, we're going to delete it. And as you notice, as that one was deleted, we have a significant consequence in terms of the what's called the reading frame. So we've gone from our particular sequence and we've now shifted all of the nucleotides that existed upstream of that particular, or sorry, downstream rather, of that particular deletion. And so the reading frame has altered, okay, because of what we call that frame shift. The consequence of that is our amino acids that we code for beyond that particular point also then change. So we've now got a different set of amino acids coded for beyond that particular deletion. Okay, so the deletion occurred here. We've got a frame shift, so that's lead to anything downstream of that particular change now coding for a completely different amino acid. So what do you think the effects of that are going to be? Again, pause the clip, have a think, and I'll quickly talk through kind of some of the consequences. So with insertion or a deletion, depending where it is, if it's right near the beginning of a gene, it have an impact on the whole sequence beyond that particular insertion or deletion. If it occurs towards the end, it may not have as huge an impact, but it is going to lead to the incorporation of different amino acids and therefore have a huge impact. Right, for the next task, what I'd like you to do, it may be worthwhile doing the screen grab of the particular genetic code I'm going to put up on the screen, because I want you to use the letters at the right-hand side. You're going to practice a little bit at doing some of these genetic code shifts and see if you can figure out what the consequence is. So on the next slide, if I just kind of click through, our next slide has a slightly different version of the genetic code to the one I've given you previously. Interpreting it is fairly straightforward though. Okay, you're looking for your first letter, so head over to this side and find the first letter of your genetic sequence, and then you're going to look for the second letter and so on, and from that you can determine what the particular amino acid is. So if we had AUU, this would lead to this particular code down here. Okay, if I just zoom in and focus on that, it's going to lead to that particular section, ILE. We're then going to go over to the side and we're going to look to find ILE, which stands for isoleucine. Okay, when we get to isoleucine, what I'm interested in you working out is the single letter code that goes with isoleucine. So as you get the next sequences, what you need to do is work out the amino acid that is coded for, then work out the individual letter, and you need to make a note of the letter that goes with it. Okay, so let's have a look at what we mean by that. So hopefully take a screen grab of this particular screen, put that into another document, print that screen grab off, and then hopefully you can work through the next particular task. So here's an example. This is our particular DNA sequence, so I need you to transcribe that DNA sequence into messenger RNA and then translate that messenger RNA into an amino acid sequence using the single amino acid letter code it will spell a message. So I would recommend you pause it there and can you note down that particular message. Hopefully the screen at the top left is big enough for you to be able to read and work out the code. So I recommend you pause there. Okay, so in terms of transcription, you should have come up with the following, all being well, as it tries to click through. So you should have come up with, okay, that particular sequence. And if you've got that particular sequence, let's take you through it. So we start with AUG. So you'd go up to the top and you'd find AUG, and you can see, as I've said before, AUG is always the first amino acid. It's going to code for the amino acid methionine, okay, which has a three-letter code MET. If we then go across to methionine and look at the individual letter code for methionine, I'll just zoom in very briefly on that, hopefully. Let's have a quick look. Okay, so if you have a look at methionine and zoom in, you can see that methionine has the particular code letter M. So I'm going to zoom back out. And so if we go through each of those particular triplets and look at the amino acid that it codes for, we would have the following message. Hopefully you managed to get this message. Okay, and you should have, if you read it through, mRNA is great, and end up with a stop code on. So now you've got that first particular sequence, you can use this basic principle to then do some similar tasks on the next few slides. Again, I'll put the screen up, pause after each one, and then see if you can determine what the impact would be in terms of the amino acid sequence that is generated. So here's your next one. So nonsense mutation, and think what's going to happen to the DNA sequence, how would this affect the protein? Okay, pause the screen, see if you can complete that particular task. Okay, that's what you should have come up with in that particular sequence. And let's look at the amino acid sequence. Okay, immediately we didn't get any further than the stop, it went MR stop. Okay, so if we kind of zoom in and look at why it went stop, we're looking at UAA. Okay, so we're going to look in here, in fact, there's the U. Okay, we go across and we can see that UAA leads to our stop instruction at the top here. So that particular amino acid sequence would not be translated any further. Let's look at the next one, okay, and see what the consequence of that would be. This is our missense mutation. Okay, pause the screen. Hopefully you managed to transcribe that okay. And let's look at the translation of this one. Go to the amino acid sequence. Okay, it's a minor one in this case in terms of what we can see, but maybe the consequence of that would be more significant. So we've got mRNA, we had is great before, we've now got a different amino acid, okay, AGA. Let's just have a look, see where that one came from. Okay, so we're looking for AGA, so we'll go through the sequence, we find AGA, and we can see it's arginine. Okay, so ARG in terms of AGA, if we go to the top, that led to the amino acid single letter code of R. So instead of that mRNA is great, we ended up with mRNA IR great. Depending where that is, that could affect your secondary structure, so it could affect your production of double helices, helices within the structure, could affect the production of beta-pleated sheets. Certainly it's going to impact the tertiary structure, how that particular sequence would fold up, and an impact on the functionality of the protein that you produce. Let's look at the silent mutation. This one should be fairly straightforward, I hope. Pause the screen. And here's our silent mutation. Here's the amino acid sequence. It hasn't changed. So even though we've got a base change, we've still got mRNA is great in terms of the amino acid sequence. So that silent mutation, because of the degenerate nature of our amino acid code, or our genetic code I should say, we've now got no change in the amino acid sequence that we have, but the original DNA sequence has altered. Now this next one might take you a little bit longer to do. Okay, we're now going to look at the impact of a deletion mutation. So there's the sequence. So again, pause the screen and then do your transcription step, and then do your translation, and let's have a look at the consequence. Okay, so there's our transcribed sequence, very different to what we've had before. And as we go through and look at our translated sequence, our amino acid code, we no longer have the message we had before. mRNA is great. We've now got MLM, LLV, a completely different set of amino acids. So that original protein that we produced, we're no longer manufacturing. So we have changed it quite dramatically in terms of its functionality. And it would probably be a completely non-functional protein. Okay, so let's have a look at some of the mutations. We've got things like chromosomal mutations. So we've got this idea of what happens with a gene deletion. As it sounds, it's not just a single base, it's the whole gene that can be deleted. Very straightforward, the gene's gone. Okay, so depending where that occurs, it can have significant impact. If that was in a production of a gamete and you no longer have that particular gene, and that gamete was then used in terms of fertilization, the new organism, that zygotic cell originally that you produce, wouldn't have that particular gene from one parent. And let's have a look at gene duplication again, as it sounds. Okay, we've now got, in this case, an extra couple of genes that could impact on production of proteins in terms of quantities of proteins produced. But again, it is a fairly straightforward chromosomal mutation. There are others too. We can have something called inversion. Inversion, we're kind of changing the order in which things occur. That might affect what is actually transcribed. And translocation. Translocation is about moving particular sequences from one chromosome to another. So in this case, we're swapping a couple of them over. Again, those can have a significant impact too. We've also got other well-known ones, things like whole chromosomal mutations. So in this case, this is where an entire chromosome can be lost during cell division. The well-known one, if you look down and have a look down at the bottom for chromosome number 21, in this case we've got 3 copies of it, and the genetic disorder Down syndrome is caused by having that extra chromosome 21. So we've looked at the effect of many of these mutations. Let's kind of try and summarize some of those key ideas. Effects can be as simple as it could produce a better protein. It may be that amino acid sequence means that particular enzyme that has been produced is more effective or works better at different temperatures or a different pH. So that could be an advantage for the particular organism with that mutation. It might have no effect at all. If it's a mutation that doesn't change the sequence, or even if it does, it might not impact whatsoever in terms of what you produced, so it makes no difference. However, some mutations are also can lead to production of an inferior or production of no protein, and that may be fatal. It could cause disease or other consequences for the organism. So there is a kind of a range of different consequences for mutations. Key thing for us to remember is focusing on the impact on things like tertiary structure and changing that sequence of amino acids and what the consequences of that would be. So what can cause mutations? Quite simply, things that we can have that can lead to mutations are things like anything that's classed as being a mutagen, something that is mutagenic. So X-rays, ionizing radiation, various different chemicals, all of these things can have an impact on whether mutations will occur or not. Right, your particular task. This is kind of what you're going to do within the next lesson. This should take a little bit of time. So this is the key theme. I want you to go and look into impacts of some particular mutations and particularly find out about what's called non-disjunction chromosomal mutations. 1,500 word assignment, that's a guide, okay? I certainly don't want anything longer than that. I think most students usually do it in between about 800 to 1,000, so don't sit and do a word count, just produce what you need to produce. I'd like it fully referenced. Minimum, you should include a description and explanation of, I want to see Down syndrome, I've already briefly mentioned that one, Turner syndrome, and then find something else you think I might be interested in. I've seen quite a few over the years, so I'll be intrigued what you find. I don't want you cutting and pasting. That's not a review. If you use any diagrams or anything you've got, please reference those as well and label appropriately. If you are referencing, think about the technique for referencing. So there's a couple of examples in there how you should reference. So in the body of the text, having the author's name and the date will be sensible, and then at the end of your article include your reference list. Particularly for websites, I'll expect nearly everything you find at this stage is going to be a website, because you've got very little access to anything else. Just make a note of the kind of date that you access that particular piece of information. Okay, hope you enjoyed that. That's it, there's a task. Please email me anything that you need, and I'll try and put another lesson together. Hopefully it might be a little bit better than this one, for later in the week. Okay, see you soon, guys.